Hey team, Dr. Jack Gordy, and in the previous videos I've been discussing how neuroinflammation contributes to Alzheimer's disease, how the activation of microglia causes them to damage neurons directly and also recruit in damaging immune cells like neutrophils, which also then go on to damage the neurons directly. But how do the neutrophils know that the amyloid is there? How do microglia detect amyloid in Alzheimer's disease? Now that's a super interesting question. Now another way to phrase that is, what are the pattern recognition receptors for amyloid? Now we know microglia surveil the extracellular environment using receptors on the surface of their membrane or receptors within their phagosomes or even cytosolic receptors. These are receptors that float around their cytosol to detect pathogens in the or damage-associated molecular patterns in the extracellular environment. So they're surveying the environment using these receptors that we call pattern recognition receptors to say, is there any diseases or is there any damage in the extracellular environment? Now, these are called pattern recognition receptors. So which ones of these pattern recognition receptors are involved in amyloid? Now, this took us a long time. This was a lot of research to figure out this process, and there was a lot of rabbit holes and a lot of dead ends in this research. And that's because there are tons of pattern recognition receptors. There are the toll-like receptors, the nod receptors, the RIP kinases, for example. We've also got the lectin receptors. These detect carbohydrates like on fungus. And then there are the inflammasomes. These are cytosolic receptors that respond to a number of different stimuli. There are a lot of pattern recognition receptors. I don't want you to learn them all, but which ones are involved in Alzheimer's disease? Now, I'm going to tell you basically what we've sort of concluded, and then I'll jump into some of the research and how we discovered this. Okay, so basically we think the primary signal is this signal here. It requires 3 receptors on the surface of the membrane: CD36, toll-like receptor 4, and toll-like receptor 6. And we think basically this is what's going to happen. We've got the amyloid up there. It's going to bind to CD36. CD36 is the binding and recognizing motif that stands on your that sits on your outer membrane, but it doesn't have the sort of active secondary signaling molecules going on inside as a receptor. It's just bound the CD36. Now, what we think happens next is that it forms a heterodimer. Now, a dimer is 2 molecules, 2 receptors binding together. So that's TLR4 and TLR6 binding together. And hetero means different. So if it was TLR4 with TLR4, which does happen with LPS, then that's a homodimer. But with hetero, it's TLR4 and TLR6 forming a heterodimer. Now, when these two get together, they activate a kinase cascade on the inside. And a kinase cascade is one enzyme phosphorylating another enzyme, which phosphorylates another enzyme, which phosphorylates a transcription factor, which causes an upregulation of genes. And it's how so many receptors work, some sort of kinase cascade. Now, this uses a famous intracellular part called MyD88. Don't worry about that, but that's just so it pings in your ears if you hear it again. So we get a MyD88, which is an internal kinase on that receptor, causing a phosphorylation cascade. And the end result is the phosphorylation of a protein that inhibits NF-kappa B, and when that inhibitor is phosphorylated, it releases NF-kappa B and allows it to go into the nucleus. Now, when NF-kappa B goes into the nucleus, it acts as a transcription factor, binding to promoter regions of the DNA and facilitating RNA polymerase to come in and cause the expression of a whole bunch of genes. And these genes are inflammatory genes. They're inflammatory cytokine genes, but they're also enzymes that produce reactive oxygen species, which allow the microglia to attack whatever pathogen it thinks it's been exposed to. And it's also enzymes that cause the production of inflammatory signaling molecules like prostaglandins. So that's the primary pathway that amyloid interacts. Now let's have a look at the research there. Now basically they took microglia and exposed them to amyloid. And then they genetically engineered the microglia to see how could we block this response, right? So you could imagine maybe it's NOD1, which is an inflammatory pattern recognition receptor. So you genetically delete NOD1 and you notice that you don't change the response of the microglia to the amyloid. Therefore, you know it's not NOD1. So you can go through a whole bunch of these genes until you start to figure it out. So this is what they figured out. It's quite peculiar, really. If you knock out TLR4 from the microglia, you block the response of the microglia to the amyloid. Now, they also measured inflammatory cytokines, but in this measurement, they're measuring reactive oxygen species. So when microglia are activated, they spit out reactive oxygen species like peroxides, like hydrogen peroxide, in an attempt to kill the pathogen that they think that they've detected. So should a pattern recognition receptor detect a pathogen, they want to release those reactive oxygen species like peroxide to kill the pathogen. Hydrogen peroxide, you can imagine, kills pathogens. It kills everything, but it kills pathogens. So if you measure how much hydrogen peroxide and other reactive oxygen species are coming off the microglia, you're measuring how active they are, really, how inflamed and activated they are. So they've got reactive oxygen species on the y-axis here. The control microglia aren't producing any. The amyloid-treated ones are producing loads. So here we can see lots of reactive oxygen species being detected. And when they deleted the toll-like receptor 4, they blocked the response. Now, an inferior researcher might go, boom, job done. It's all through toll-like receptor 4, but they actually wanted to check a whole bunch of other ones. So they also noticed when you delete TLR6, you block the whole response completely. And again, you might go, okay, job done. But then they also showed that if you delete the CD36 receptor on the cell surface, you completely destroy the whole response. So now you can see where that model came from. If it was working through TLR4 and TLR6, you would expect deleting one or the other to maybe reduce the response by 50%. But, TLR4 or TLR6, you would expect deleting one to maybe just reduce the response by 50%. But deleting either of them reduced it by 100%, kind of showing that you need them both together, so forming a heterodimer. You need those two receptors coming together in order to get the kinase activation and the production of those reactive oxygen species. But then they showed CD36 was essential too, and they think CD36 is doing the binding of the amyloid and then bringing together TLR4 and TLR6 into a heterodimer, and they showed that with a bunch of other experiments. And here it is here, but this time with an inflammatory cytokine, interleukin-1. Now, see if you delete TLR4 or TLR6, you block interleukin-1 production. And here they're blocking MyD88. Now, I mentioned MyD88, that internal domain that's essential for the kinase cascade. So they're demonstrating that TLR4 and TLR6 are working through MyD88 as a kinase cascade. And that's because TLR4 can sometimes work with other kinase cascades, such as TRIF. And it depends on whether TLR4 is on the cell membrane or within a phagosome. And when TLR4 is facing into a phagosome, then it tends to use TRIF as its internal kinase signaling molecule. So this is saying the microglia doesn't need to eat it. It can just have it on its cell surface and will get MyD88 kinase, dependent kinase, production of interleukin-1 beta. So this is mRNA of interleukin-1 beta. Very cool experiment showing essentially you need all three of those cell surface receptors to activate the kinase cascade and induce IL-1 production as well as reactive oxygen species. So IL-1 is an example of a cytokine that's an inflammatory signaling protein molecule, but also there are other molecules involved in the inflammatory response, and one example is prostaglandins. So let me jump into those as well. So prostaglandins are a small molecule. They're produced by a COX enzyme that digests the phospholipid membrane. It digests some of the phospholipids within the membrane to produce this prostaglandin. So COX-2 is an enzyme that digests the membrane into an inflammatory signaling molecule. Now, an important point of that is because it came from a phospholipid, it's actually a lipophilic molecule. It doesn't like water. It likes lipids. Now, what's unique about that compared to, say, a protein cytokine is that it means it doesn't really want to flow in the blood. It wants to diffuse out through all the membranes of the cells and through the fatty tissue. And what that is great for is getting through barriers. It'll get through any barrier. So the blood-brain barrier, for example, blocks most water-soluble molecules from getting from the blood into the brain. Prostaglandins prostaglandins diffuse out, and it creates that local response. Now, prostaglandins also activate pain-sensing neurons, which is why prostaglandins make it sore. It's why often inflamed things are sore, is one of the major reasons is these prostaglandins. Now, this COX-2 enzyme is controlled by NF-kappa B. So we know that if you activate TLR4 and TLR6, you activate NF-kappa B, which causes the expression of this COX-2 enzyme, which digests the phospholipid membrane, just a little bit of it. It's not a major breakdown of the membrane. It's just taking some of the components of the phospholipid membrane and turning them into prostaglandins, and those prostaglandins then go on to act on prostaglandin receptors to cause inflammation as well as pain. A COX-2 enzyme can be turned on by a DAMP or a PAMP, such as through TLR4 activation. It can also be activated by cytokines. So if a cell has an IL-1 receptor, for example, if you activate that IL-1 receptor from using an IL-1 cytokine, an interleukin-1 cytokine, you'll get COX expression as well. Now, it's slow and capable of prolonged signaling, and it's also quite local. It doesn't flow around the blood. It's more gently diffusing out through the membranes, and that's because of the lipophilic nature of it. Now, non-steroidal anti-inflammatory drugs. These are pain-relieving anti-inflammatories that you buy off the shelf at the pharmacy, such as aspirin and ibuprofen and Voltaren, diclofenac, mefenamic acid. There's a whole bunch of them. But all the famous pain-relieving drugs that you can buy over the counter basically are non-steroidal anti-inflammatories, and their main job is to inhibit the COX-2 enzyme. They also inhibit COX-1, some of them, and that's a complicated relationship, affecting your stomach lining and blood clotting and all sorts of other things. But the main goal is to inhibit COX-2, which is that real inducible enzyme that's associated with pain as well as inflammation, and that's all through prostaglandins. Yeah, and so its primary regulator is NF-kappa B. So the COX-2 enzyme is out here. NF-kappa B causes the mounting of RNA polymerase all over the DNA, and one of those genes is the COX-2 gene that will now turn on in response to NF-kappa B activation. Woo! Okay, next up we have cytokines, just to explain some of the differences. And they are again caused by PAMPs and DAMPs activating receptors, or cytokines activating receptors, often causing NF-kappa B activation, and then that will cause gene expression. Now, sometimes cytokines require post-translational modification. So interleukin-1 and TNF-alpha, for example, aren't produced ready to go. And they require sometimes a second signal, and that second signal will cause their maturation. They'll go from an inactive pro-molecule to an active mature molecule that's now released out of the cell. But there are other cytokines that are just automatically released. So they would have a signal peptide that would send them to the rough endoplasmic reticulum, which would then send them to be secreted straight away. Interleukin-1 does not have a signal peptide, so it cannot get secreted straight away. Okay, cool. Interferons in that group too. And a lot of cytokines, not all, but a lot of cytokines are regulated by NF-kappa B, for example. So again, you turn on NF-kappa B, it'll go in and cause the expression of IL-6, IL-1, and TNF-alpha. So they are proteins, which means typically they're water soluble, which typically means that they travel around the blood, but they can sometimes struggle to cross barriers. So they're not like prostaglandins, which are lipophilic. They're more hydrophilic, so they spend a lot of time in the blood or in the fluid compartment. That means that they can signal long distances. So they are also slow and capable of prolonged signaling. So there are faster ways to induce inflammation, but the prostaglandins and cytokines are a little bit of a slower way to do it. Right, so DAMPs, for example, damage-associated molecular patterns, are a much faster way to induce a local inflammatory response. Okay. Histamines are also incredibly quick. Anyway, okay, so they're massively varied. So cytokines can be anti-inflammatory, and IL-10 is a really good example of that. Interleukin-10 is an anti-inflammatory cytokine. But these ones I mentioned here, IL-6, IL-1, and TNF-alpha, are pro-inflammatory cytokines. And chemokines, I've mentioned this before, are a subset of cytokines that attract up a concentration gradient. So they are proteins that attract cells up a concentration gradient. Okay, so why is TLR4 and TLR6, which are normally more associated with pathogens, such as lipopolysaccharide from bacteria. Why are they activating in response to amyloid? And that's a confusing thing, because amyloid is a protein we've produced. It's not really a damage-associated molecular pattern. There's no damage. Amyloid doesn't necessarily correlate with damage. It's just a cleavage product of a normal synaptic protein, amyloid precursor protein. So why are microglia receptors detecting this? And it's an interesting question. So I'm going to explain to you a technique that might help reveal it, right? So these are bacteria. They've been streaked out on an agar plate, and they've been stained with a stain called Congo red. Now, Congo red binds to amyloid proteins, and Congo red was first discovered as a bacterial stain to stain amyloid proteins and amyloid-like proteins that are expressed on bacteria. So we can see that these bacteria are testing up positive under the Congo red stain, showing that these bacteria have amyloid-like proteins protruding from their cell wall. Now we can use that same Congo red stain to stain Alzheimer's disease. So here, this is an Alzheimer's brain, and in red here, we have the plaques. Now the amyloid plaques are staining with a stain that we normally use for bacteria. And this might be your first hint. Perhaps these amyloid oligomers and amyloid fibrils are molecularly very similar in structure and properties to proteins that project from the cell wall of many pathogenic bacteria. So perhaps it is an example of molecular confusion. Perhaps our receptors that are imperfect, they've just evolved to detect bacteria, happen to also detect amyloid and amyloid fibrils. And I want you to think about this in terms of evolution, right? Alzheimer's disease typically hits people at the age of 70. That is well after their reproductive period, well after their nurturing of siblings period. And for most of human history, having a negative effect on a 70 or 75-year-old survival, such as Alzheimer's disease, would not have a strong evolutionary pressure. There's not a lot of reason for these TLR4 and TLR6 receptors to evolve not to detect our amyloid. Especially when you consider one of the other threats, which is bacterial infection. They need to detect those bacteria because that will kill you as a baby, that'll kill you as a 10-year-old, that'll kill you as a 20-year-old. Right when it's very important from an evolutionary perspective. So the evolutionary pressure is for us to make mistakes. It's better to be sensitive and not miss a bacteria and occasionally mistake a protein that's associated with old age as a pathogen than it is there's not much of a selection pressure to avoid detecting our own amyloid that builds up in our later life, right? So our receptors are probably just hypersensitive to amyloid-like proteins, and there's no pressure for them to figure out it's not a good idea to respond to these amyloid plaques as if they're pathogens. Evolution has not prepared us for that. Okay, so that's just a theory, but it's a reasonable theory, but it is just a theory about why our pattern recognition receptors that are designed to detect bacteria typically have evolved largely to detect bacteria are now detecting a non-bacterial protein that we produce. That's possibly why. Okay, so does amyloid look like a pathogen, essentially? However, this might be a long video, sorry guys. But what about amyloid fibrils? Now, these are the plaques, right? These aren't soluble, so they're not floating around in the fluid, so they're not available to sort of bind to receptors, right? And amyloid fibril is really going to struggle when it's not sitting in solution to bind to a small protein receptor on the surface of the membrane. Also, they're often as big or about half the size of a microglia, so they're not going to be binding to a little receptor. Something else is going on there. So how do the amyloid fibrils activate the immune response? And that's when we come into some of my research. This has been, 7 years, 8 years of my life so far has been researching this topic. And it's this topic of these proteins called inflammasomes, in particular one called NLRP3. What it stands for, it doesn't really matter. A lot of genes don't really mean anything, their names. It's not like receptor pyrene-containing protein 3, okay? But it doesn't matter. It's the NLRP3. Everyone knows it as the NLRP3 and Alzheimer's disease. So this is the NLRP3 inflammasome. Now, each one of these, just one molecule here, is an NLRP3 protein. That's the receptor. That's the pattern recognition receptor. It's an NLRP3 protein, and it's oligomerized into this thing that we call an inflammasome. And that's when it's activated. It all sticks together in this ninja star-like protein. Gosh, it's a beautiful molecule. You should really all just research this. Look how good looking that is. And it forms this complex when it's activated. Now, there are links to NLRP3 and Alzheimer's disease. So let's jump into that now. Okay, so it all ties into interleukin-1 beta. I've talked about interleukin-1 beta as one of the most inflammatory cytokines we've got in our body for almost every video on this channel, because this is what I research, interleukin-1 beta. But the story of interleukin-1 beta is quite complicated. So here we have NF-kappa B, and it's bound to the DNA, it's mounted the RNA polymerase, it's caused the expression of the gene of interleukin-1 beta, and it's now being produced. However, it's produced as an inactive form, pro-interleukin-1 beta. It's not active, okay? It's a large molecule that's not active. Essentially, it's got a big protein stuck onto it to inactivate it. It's like if you have a key and then you weld a big chunk of metal to that key now cannot fit into the lock and activate the lock. So it can't activate the receptor. It's got a big useless chunk attached to it. So we need to chop that off before it's now mature interleukin-1 beta. So we chop the chunk off, and now the key will fit in the lock and open the receptor. So we need to chop it off in order for it to be activated, and indeed in order for it to be secreted. Now, it's chopped by an enzyme. This enzyme is called caspase-1. This caspase-1 actually chops a whole bunch of proteins, but one of the proteins it chops is pro-interleukin-1 beta, and it converts it into the mature form by chopping the useless chunk of protein off, called the pro domain. Caspase-1, however, is also produced in inactive form. It's a pro-enzyme, a zymogen, if you will. And so it needs to be activated too. That's when my baby, the NLRP3 inflammasome, comes in. So it normally sits as a monomer in the cytosol there. Then it detects a disturbance in the cytosol. Something changes in the cytosol, and that will activate that NLRP3 receptor. It will then oligomerize into the ninja star that's called the inflammasome, and that will bring in an adapter molecule called ASC. And it actually brings in so many ASC adapter molecules that it becomes as big as a mitochondria. It's so huge. It's just a protein complex, but it's massive, and that's why we call it an inflammasome, because it looks like, a lysosome. It's as big as a mitochondria. It's like 2 microns wide. It's huge. Each of those S molecules can recruit a caspase and activate the caspase. And it actually forces two caspases together to activate themselves. It's called autocatalytic, self-catalytic. So the 2 caspases get brought together by the ASC molecule, and it activates, and you end up with active caspase-1, which cleaves the pro-IL-1 into mature IL-1. Super, super cool. And this is called the inflammasome complex. And this is that post-translational modification I mentioned when I talked about cytokines. So sometimes cytokines are produced in an inactive form. They require some sort of post-translational modification, like cleavage, in order to become active. And you might ask, why have this 2-stage thing? Well, interleukin-1 beta is one of the most inflammatory cytokines in your body. If I inject it into you, you will immediately get a temperature. If I inject too much, you'll probably die from leaky blood vessels, from too much of an immune response, too many activated neutrophils, drop in blood pressure, all that jazz, right? So it's kind of a nuclear bomb. And like a nuclear bomb, it's good to have 2 keys that need to be turned in order to launch the nuclear bomb. So the expression of interleukin-1 and the expression of NLRP3 is one key, and then the activation of NLRP3 is another key in order to get the release of interleukin-1 beta. So that's why I think there's those 2 barriers, because you don't want to accidentally turn on your interleukin-1 system. It's going nuclear when it comes to an innate immune system. Right. But it gets more complicated. Interleukin-1 beta still can't get out of the cell, really. There are some subtle ways, but for the most part, interleukin-1 beta, mature, struggles to get out of the cell. So we need something to happen. So you should all be familiar with apoptosis, which is programmed cell death. The cells bleb, they pop out phosphatidylserine, and they also send out signals to the microglia and the macrophages, come eat me, and they get eaten. There's necrosis, which is uncontrolled cell death. That's like a squashing or a popping by a toxin, and it means that the cell's gone so dysfunctional that it can't die in a safe way, and it just releases everything. It's gross. Next, we have pyroptosis. Now, pyroptosis is when an immune cell fills itself up with cytokines and then pops. And when it pops, it releases the cytokine, a huge amount of it. And that's what happens with interleukin-1 beta. It's called pyro because interleukin-1 used to be called a pyrogen because it caused your body temperature to go up. It causes you to get a fever. So it's called pyroptosis because it's a fire. Pyro, pyromaniac, it's to do with fire, it's to do with temperature. It's an inflammatory way to die. Pyroptosis, super inflammatory, it's a fiery way to go. It's pretty cool. So that's what happens with that's how interleukin-1 typically gets out of the cell. There are other ways, but for the most part, it happens through pyroptosis and the cell popping and releasing interleukin-1. So let me now go step by step on NLRP3 and amyloid, because it's a bit more complicated than what I've explained, even though it's super complicated what I've explained. But I've got to tell you about this because this is amazing. Okay, so here we have fibrils, oligomers, and monomers of amyloid. Right, as I've already discussed, oligomers and monomers have the ability to bind to the CD36 and to cause the heterodimer of TLR6 and TLR4. So the monomers and oligomers are doing this. They're activating these receptors. These receptors, once activated, will then cause the inhibitory molecule of NF-kappa B to come off because it will get phosphorylated and stop inhibiting it. It's called IKK. So that will come off after being phosphorylated. And now NF-kappa B will go in, bind to the DNA, and it will mount RNA polymerase, and it will cause the expression of lots of things, including things like IL-6, TNF-alpha, but also pro-IL-1 beta, pro-caspase-1, and NLRP3. ASC was already being expressed over there, and ASC is kind of constitutionally expressed in the cytosol. So now we've got NLRP3, pro-IL-1, and pro-caspase-1 in the cytosol ready for their activation. We need that second step now. So next we have oligomers and fibrils binding to phagocytotic receptor complexes. Basically, the membrane's not great at just taking stuff up. It often needs receptors on the surface there to help take up fibrils and oligomers. And it will now bring it in as a phagolysosome. But there's a problem, especially with fibrils, but also oligomers. The phagolysosome can struggle to digest what's inside it. And actually the amyloid can end up bursting the lysosome. So the phagolysosomes can end up rupturing. And when that happens, digestive enzymes come out and we end up with a lot of problems. So the phagolysosome has now ruptured. And what this can actually do is it can compromise your membranes. It can activate receptors, and it can also activate enzymes that create pores that form in your membrane, and it can actually damage the membrane itself. So now the membranes become leaky. We end up with potassium efflux and chloride efflux. Now both of these things are essential, so potassium and chloride rush out of the cell because of receptors as well as pores being formed in the membrane. And that potassium and chloride efflux causes NLRP3 to oligomerize into its inflammasome. So the potassium and chloride efflux has caused NLRP3 activation there. Once we get NLRP3 activation, we get the caspase-1 activating. And the caspase-1 cleaves pro-interleukin-1 beta into mature interleukin-1 beta. However, the mature interleukin-1 beta still can't really get out of the cell all by itself. It needs something else to happen. So what happens is active caspase-1 goes on and it activates another molecule. Now this molecule is called gasdermin D. Now gasdermin D, once activated, can form a pore in the membrane. So it's inactive, it needs to be cut. Once it's cut, it oligomerizes into a pore, and that pore right there can allow the mature interleukin-1 beta out of the cell. It can also kill the cell, and that's pyroptosis. So the pores forming from the gasdermin D causes pyroptosis, which is very, very cool. And I understand it's a super complicated pathway, but it's super important and it's so fascinating. There's even a million more other things you could explain about it. Biology's kind of fractal. You could zoom in on this one bit, say here, and it gets infinitely more complicated. NLRP3 is bind to endolysosomes, it's ubiquitinated, it must be deubiquitinated, it must come off the it must come off the endosomes, and blah blah blah. There's a million things. There's a NEX7 and there's another molecule. Any bit within here you can zoom in on and it will be super complex. That's why biology is fractal. You zoom in, it stays complex. It's amazing. It's a never-ending well of research if you want to jump into it. Okay, so this is the gasdermin D. So here we have it, and it's inactive. It's got this orange chunk bound to it, which deactivates it. You cut it, and now it will insert itself into the membrane and will form these beautiful pores in the membrane. And IL-1 can now come out of it. There's some evidence that the pore is a little bit selective, and it will mostly let IL-1 out. And not let some other things out. But eventually enough pores can form in your membrane that the cell will pop. And then everything comes out, and that's called pyroptosis. All right, so how do we know NLRP3 is important in Alzheimer's disease? Well, you might have seen this experimental paradigm before. It's very common. You get an NLRP3 knockout mouse. This mouse has no NLRP3 receptor. You cross it with an Alzheimer's mouse. This is an APP-PS1. It's got 2 familial mutations that we know cause Alzheimer's disease. One in the APP protein, one in the gamma secretase. And what happens is you cross those and you'll end up with, after a few generations, wild-type mice, NLRP3 knockout mice, Alzheimer's mice, and Alzheimer's mice without NLRP3. Right, okay, so now we've got these mice. Let's test them. What happens in the Alzheimer's disease? So we use the Morris water maze again. So these mice have been trained to find this platform over 5 days. Then we remove the platform and see where the mice swim. Now, a mouse with a good memory goes, whoa, I'm pretty sure. So we're looking down upon the maze and we've tracked the mouse, the trail of the mouse. And here's the hidden platform there, and this is the water maze there. So we've trained it to find this platform, and it's just swimming around that platform. It's like, I know the platform is here, I know the platform is there, but I can't find it because we've removed it. But we know that this mouse has remembered where the platform is over the 5 days because it's looking for it where the platform should be. This is an Alzheimer's mouse. It can't remember. So it's just doing a general search pattern. It's like it has forgotten the previous 5 days of training. It's got a poor memory. It's forgotten all its training, and it's just swimming round and round in circles. It's got no idea where that platform is, despite it's been trained for 5 days to find it. Here is an Alzheimer's mouse that's NLRP3 knocked out. So it's not releasing the IL-1, NLRP3 is not being activated, so it's not having that inflammatory response. There's probably no neutrophils coming in, there's probably no microglial phagocytosis of neurons going on, no release of reactive oxygen species, none of that kind of stuff's going on. And what we can see is the mouse has a perfectly good memory. So if you inhibit NLRP3 activation in Alzheimer's disease, you delay, perhaps prevent the amyloid-induced cognitive deficits that we see in these animal models. And this is just the training data. So this is the mice getting faster and faster and faster. They're traveling less distance. This is actually a measure of distance. So they're traveling less distance to get the platform. These guys were actually trained for 8 days. And in black and blue, we have wild type and NLRP3 knockout mice. In green, we have an Alzheimer's mouse that is also NLRP3 knockout. And in red, we have an Alzheimer's mouse. So in red, the Alzheimer's mice pretty much don't get better throughout the whole 8 days of training. Whereas in green, the Alzheimer's mice that are NLRP3 knockout do learn over their 8 days and get as good as the wild type mice. They also measured the amount of mature interleukin-1 beta in the brain, which is a very hard thing to do. So that's super impressive because you can see it's femtograms, tiny, tiny amounts there are being released. And we can see we've got mature IL-1 in the brains of Alzheimer's mice and no mature IL-1. Same levels as the wild type, basically, in the NLRP3 knockout. So we're blocking IL-1 release and we're getting better cognition in these mice that are NLRP3 knockout. This leads to the hypothesis of a paper that I'm about to break down. It's my paper, which is that factors which influence NLRP3 activity and IL-1 secretion will affect Alzheimer's disease progression. I've got 3 big papers on this topic. I've got 2 papers on can we inhibit NLRP3 as the therapeutic target for Alzheimer's disease. One's in animal models and one is in looking in human data, which is very cool. And over here is, what about, can we sensitize NLRP3 through environmental factors that accelerate Alzheimer's disease. And I mentioned this in the next video. I look at zinc deficiency because we know zinc deficiency sensitizes NLRP3. We also know it's one of the most common malnutritions in the world. So perhaps if you're zinc deficient, you've sensitized NLRP3, you're going to have worse Alzheimer's disease. And that's what I show in a paper. And in that paper, I use human data, mouse data, and cell data. I'm very proud of that paper too. I'm not going to break that down though. I'm going to focus on inhibiting NLRP3 as a therapeutic target of Alzheimer's disease in the up and coming videos. And that's the paper right there. That's what we're going to break down. It's using NSAIDs to inhibit NLRP3 to protect against Alzheimer's disease. Oh, thanks team. Next video is on Western blots.